All right, great. Thank you so much. And thank you for inviting me to speak today. I've really been looking forward to this water cooler chat. So I teach biology, biology 1, general biology 2, and microbiology, sometimes all in one day, and absolutely love it. In the spring of this year, I participated in a faculty mentoring network. And before that, I hadn't heard of the 4D framework. But it was an enriching experience to discuss the framework and receive guidance on how to apply it to my courses and then create teaching resources for others. One thing I found most interesting was that many of the lessons and activities that I had already developed could easily be adapted with just a slight shift in perspective, incorporating elements of the 4D framework to truly enhance the student experience. So I'm really excited today to share with you how the 4D framework has influenced the integration of ecological concepts into my biology and microbiology courses. So I feel as educators, it's essential that we not only teach our students the core scientific content, but also help them develop this holistic understanding of ecology, really enabling the student to apply the ecological principles to real world challenges. Let me see. All right. So I believe most of you probably already know what the 4D framework is, so I'll keep this introduction brief. The Four-Dimensional Ecology Education Framework was developed by the ESA as a structured way to integrate ecology into education across multiple disciplines. It serves as a fantastic guide for embedding ecological principles into courses that traditionally might not explicitly focus on them, like microbiology. For my courses, I focus on using the core ecological concepts and align them with ecological practices through hands-on lab activities. This combination not only reinforces the fundamental biological concepts, but also highlights the human-environment interactions. Additionally, the cross-cutting theme dimension offers a great opportunity to integrate the interdisciplinary topics into our discussions. So today, I'll discuss some ways I apply these dimensions in both biology and microbiology, making these courses more ecologically grounded and impactful for students. It's important to mindfully integrate the 4D framework because ecology tends to be underrepresented in traditional biology and microbiology courses, as these subjects often prioritize those cellular, molecular, and biochemical processes. And that emphasis is largely driven by the curriculum focus on the foundational concepts like genetics, cell biology, and microbiology techniques. So as a result, sometimes those ecological principles, which really connect those processes to the larger environmental systems, are often overlooked or just minimally addressed. And that imbalance can leave students with that limited understanding of how biological systems interact with and impact ecosystem, despite this growing recognition of ecology's importance in addressing global challenges like climate change, right, and biodiversity loss. So by incorporating this 4D approach, students will benefit by developing that more holistic understanding of both biology and ecology, gain the practical hands-on skills in ecological practices, then combine and fully understand the relevance of ecology to those real-world issues. So if you take a look, I included the image on the top. This shows Saccharomyces cerevisiae, the yeast commonly used in baking and brewing. So in biology and microbiology courses, students often have the opportunity to observe cells under a microscope, allowing them to examine the structures at the cellular level. And then on the bottom, that depicts a typical fermentation experiment conducted in a lab, where students mix that yeast with juice, cover the flask with a balloon, and as the yeast consume the sugars, they produce ethanol, and carbon dioxide is released, captured in the balloon, and it expands. And that's a visual representation of the fermentation process in action. So while this is traditionally, an activity in biology or microbiology. With that 4D integration, students might explore how microbial processes link, like fermentation we have here, linked to that larger ecological or environmental cycle, which I will explain in the next slide. All right, so the first key dimension in the 4D framework is core ecological concepts, concepts fundamental to understanding how the ecological systems function. Ideas like energy flow and nutrient cycling and species interactions are critical in helping students bridge the gap between classroom knowledge and real-world ecological systems. So in the previous slide, microbial fermentation, it can be demonstrated in the lab, right? Yeast ferments the sugar, produce the gases like carbon dioxide, a clear demonstration of energy flow and microbial processes. But we can take this concept a whole lot further by considering implications beyond the lab bench. It has real ecological significance, as it also happens in nature, influencing species interactions. An example of this in action, perhaps, is the drunken monkey hypothesis, proposed by Dustin Stevens and Robert Dudley, where this hypothesis suggests that primates, like the monkey shown here, are naturally drawn to the smell and the taste of fermented fruits, which contain alcohols like ethanol produced by microorganisms like yeast. This microbial activity alters the chemistry of the fruit and also affects the behavior of the animals consuming it. The odor of the ethanol is detectable downwind since it's a lightweight molecule and it disperses readily. Over time, these primates may have evolved a preference for fermented fruits because it serves as a reliable sign of ripe fruit, which provides a foraging and survival advantage in environments where food is scarce. So by tying this hypothesis back to our lab example of microbial fermentation, we see how microbes influence species interactions and energy flow and ecosystem beyond that controlled environment of our experiments. And so this shows the students really that microbial processes, they're not occurring in isolation. They drive interactions between species and shape entire ecosystems. The next key dimension of the 4D framework is ecology practices. And the goal here is to teach ecology concepts in the field and the lab and show how students can engage with real-world environmental data. We focus on fieldwork, data collection, and analysis in the lab. And ecology practices can be effectively integrated into biology and microbiology labs. Here are two examples in the pictures. The top image shows my students conducting a survey using their quadrats, fundamental ecological technique, where students can collect data on species distribution, abundance, or diversity. This is one of my Biology 2 lab classes conducting a survey on weeds. Yes, we have lots of beautiful weeds here in Florida, right on our campus, but it's an example of how fieldwork can help students develop practical skills and data collection. While also observing plant communities in their natural habitat or unnatural habitat. And in the bottom image are some of my microbiology students. They're conducting an enumeration activity. They're working with soil samples they collected in the field, and they're performing a serial dilution to quantify the number of microorganisms in those samples. This technique helps them understand just how much bacteria is in a gram of soil. This is an opportunity to discuss microbial population sizes in the environment, which is an important skill for studying microbial ecology. So by incorporating these practices, students are not just learning about isolated lab techniques, they're seeing firsthand how biological systems function in real-world environments. And it's an integration that gives them a more comprehensive understanding of this interconnectedness of processes and that broader ecological systems. The next dimension is human-environment interactions. Many microbial processes have direct implications for both human health and environmental management, making them essential in that broader study of ecology. To give my microbiology students hands-on experience with these interactions and with applications, I take them on a field trip to a local wastewater treatment plant. Here they see firsthand how microbial processes are applied in a practical setting like wastewater treatment. The top image you see there shows a settling test, which is crucial for assessing the efficiency of treating the treatment process, measuring how well the sludge separates from the water, producing that clear effluent. And the bottom image features the clarifier, which is essential in separating water from solid particulates. And these real world observations for them, they provide the students with valuable insights into what are the role of microbes in breaking down organic material during the treatment process. And in the secondary treatment stage, the bacteria digest that organic matter, and the clarifier allows microbial flocks to settle, separating solid particles from the cleaner water before they move on to possibly anaerobic digestion stage. So this experience helps bridge the classroom learning with the environmental applications. Cross-cutting themes serve as the final dimension of the 4D framework. These themes are crucial for helping students connect and integrate various ecological concepts across disciplines, making their learning experience more interdisciplinary and interconnected. Cross-cutting themes encourages students to think beyond isolated biological concepts and instead consider how these concepts work together in the broader ecological context. Some key examples of how these themes come into play in my biology and microbiology courses can include, first, with structure and function, we look at how enzymes regulate critical biochemical processes, such as amylase breaking down starch, which can help students understand the relationship between structure and function at that molecular level. We explore the role of microbes in the global carbon cycle, methane emissions, how certain microbes can break down plastics, connecting microbiology to major environmental challenges like climate change and pollution. With systems, by studying microbial contributions to that soil health, students see how microbes are essential to sustainable agriculture, linking microbiology to ecology and also food systems. And understanding changes across time and space. This covers broader evolutionary and ecological changes such as evolution and antibiotic resistance, which helps students appreciate how biological process can vary over time and across different environments. So each one of these themes plays a critical role in linking together diverse scientific concepts, enabling students to view biology and ecology as part of this larger interconnected system. It's helpful that the American Society for Microbiology already has guidelines that align very closely with the 4D framework, which makes integrating that ecological concepts into microbiology education more seamless. Both frameworks aim to promote interdisciplinary and integrative learning that connects those core biological concepts to real-world applications. So we'll see how some of these guidelines align really well with the 4D framework. Both the ASM guidelines and 4D framework focus on those foundational processes, such as microbial roles in ecosystems and nutrient cycles. Both frameworks encourage the interdisciplinary focus to help students see the real-world relevance of what they're learning. Both guidelines, they emphasize the importance of microbial diversity and role in health and environmental balance. And a shared priority is examining how human activities impact microbial ecosystems. And both 4D and ASM guidelines promote scientific inquiry and systems thinking. So getting started with integrating the 4D framework may seem challenging, seemed a little bit challenging for me as well. But for those of you just getting started, I have Some practical strategies that I found make it a little bit easier. One is redesigning course labs by incorporating some field work, perhaps incorporate case studies using real-world issues such as antibiotic resistance or climate change impacts. Having more collaborative projects are great and help students connect microbiology with ecological concepts like microbes in that wastewater treatment. And also leveraging local ecosystems that provide hands-on ecological and microbial studies. So all these strategies provide a straightforward way to bring 4D into your classes, your courses, make learning engaging, but there is support. So to support the integration of the 4D framework in your courses, there are resources available. And part of how I got involved with the faculty mentoring network is when I was searching for the open educational resources, I came across the CUBES Hub and this opportunity. So the CUBES Hub offers a wide range of teaching modules and projects that emphasize 4D themes, energy flow, and nutrient cycling. And these resources are designed for both majors and non-majors in multiple disciplines. Then we have teaching issues and experiments in ecology. This provides a collection of classroom experiments, activities, teaching strategies, and then Course Source. That's a great platform that offers teaching resources specifically developed for undergraduate biology educators. Right. So this slide showcases the teaching resource that I developed and published as part of the faculty mentoring network group that I was a part of. This resource is entitled Exploring Microbial Diversity, an interactive in-class assignment for biology and microbiology courses. And it's available on the CUBES Hub. It offers a fresh perspective that highlights ecological concepts while helping students appreciate the vast diversity of microbes. The learning outcomes focus on helping students identify and differentiate between bacteria and archaea, categorizing the microorganisms based on their traits, and allow students to collaborate effectively. It's a comprehensive, interactive way to engage students with both ecological and microbiological concepts. And so in this activity and on CUBES Hub, you're able to explain and summarize how the activity works. And it's designed for a class of 10 to 30 students. It encourages that hands-on research, collaboration, and presentation. And each group is assigned to a specific microorganism. The group completes research on key features like morphology and metabolic requirements, but also things like key or core ecological concepts like what is the trophic level, and key ecological concepts and terminology which may not be normally used in a microbiology class setting. A feature I really love about CUBES is its flexibility. It allows users to upload multiple files, which can be freely downloaded as a package by anyone interested in using these resources. And so if you take a look, I have put on this slide some of the files that I've uploaded. The package contains instruction guides, assessments, worksheets, and sample assignments to support that activity, for example. Another example, and since I'm running low on time, I think I'm over time, I wanted to share a resource that I found that I absolutely love. I found it on Core Source, and it was developed by Mark Brodkin, and I use it in my microbiology classroom. So my students bring soil samples into the laboratory, and his resource has a package which has the student handouts, and it also has the protocol, the procedure, and all the ingredients for the setup from my lab managers. And my students get to isolate bacteria from the soil, create a pure culture, extract its DNA and perform PCR, learn about bioinformatics, And this project they do alongside their normal activities. So they get this additional research project during the same semester. So love CourseSource, use it a lot. And some activities, it's much easier to clearly integrate the 4D framework. For example, I leverage my local ecosystem by taking my general biology 2 students out on a wet walk field trip to the nearby slough, that low-lying wetland with slow-moving water. And this outdoor learning experience literally, as you see in the picture, immerses the students in core ecological concepts. They observe biodiversity firsthand, identify different species, learn about ecotones. So it's a great time, but I always consider that 4D framework. All right, and we have the benefits of integrating 4D, that interdisciplinary learning, hands-on, relevance, and inclusive learning. Overcoming challenges. One challenge I often have are the time constraints of the classes. And so when you go to the CUBES hub, you can search for activities and you're able to filter by different parameters like activity length. If you're limited, if you're looking for a lecture or test or anything like that, I absolutely love that feature about CUBES hub as well. All right, and so here's our summary. Integrating that 4D framework into these biology and microbiology courses can enrich student learning by blending this ecology with these other scientific disciplines. And ecology, as you know, is an essential aspect of biology and microbiology education. So by adopting this 4D framework, we can offer students a more interdisciplinary learning experience that prepares them to tackle ecology and environmental challenges in their future careers. And here is a final photo. Some of my happy microbiology students fully engaged in their hands-on learning approach. So thank you to the ESA, NSF, everyone who contributed to the success of this year's Spring Faculty Mentoring Network, especially to the organizers and participants that I had the pleasure of working with. Special thanks to everyone at FSW for support it takes, including all faculty, staff, and our lab technicians who help make the hands-on activities possible. Thank you to my hardworking and dedicated students and all of you here today.
Thank you so much, Amanda.
Thank you.
Thank you. So we have time for about 3 minutes, so maybe 1 or 2 questions from anybody from the audience. If you'd like to use the raise your hand, react, and we can call on you. And it looks like, Jamie, do you want to go first?
Oops, sorry, I was clapping, but I did have a question. So have you implemented a lot of this in the classroom, and what responses are you getting from students by incorporating more of the 4D network and ecology in these micro classes?
Yeah, great question. Thank you. So those 3 main examples that I pulled up, I use and have been using in the classroom, and addressing them now from a different perspective of how they're aligning with the 4D framework and how I can enhance any particular element. The first time I took students out on wet walks, of course, they were all amazed to get down actually into the slough, into the water, water up to the chest. And we're talking about ephemeral ponds and things like that. They love it, and I feel like they're more interested in it. And before, I didn't incorporate that soil lab activity in my microbiology courses, for example. Well, I just started incorporating it this past summer, and students are really engaged and feel kind of a sense of ownership, I guess, and a little bit competitive because they go out and collect their soil samples. They're instructed how to do so. They're provided the samples collection, and they have to justify, okay, why did you collect this sample here? And, you know, they're invested in the project because it's something that they've put that extra effort into. A positive response.
Awesome. Thank you so much, Amanda. That's a really good question, Jamie. Speaking of Jamie, I think it's time for your presentation if you'd like to share your screen now.
Sure. All right.
Awesome. Thank you, Jamie. We can see it.
All right. Awesome. Thank you. Yeah, well, thanks for being here today. My name is Jamie Kneitel, and I'll be talking about HEI, human environment interaction research in the classroom, focused on this cure that I developed, this human environment aquatic lab with the acronym HEAL. And this is all published on Cubes as well. And I can't speak enough to just what a great resource Cubes is. So a little bit of background. So I've been faculty for 20 years, and in 2023, I was completing my term as chair of the department, and I knew I was returning back to the classroom to do ecology. And honestly, so this was in 2023, and I had not taught ecology since 2009, and because my whole schedule was occupied by quantitative methods kinds of courses and graduate courses. And going back into the classroom and teaching ecology, I knew I wanted to take a new approach and kind of rejuvenate the class that I had taught, years ago, and really update it and with new vibrancy and speaking of cubes, one of the inspirations was, and I see James Vonesh is on this, like his whole river partners section in Cubes is excellent, and I got a lot of inspiration on that. But I saw this advertisement for the HEI Faculty Mentoring Network, and I applied and gratefully got accepted. It was a human environment interactions faculty mentoring network, specifically in the fall of 2023. And the goal of it was to incorporate the human dimension of 4D framework into a high quality educational product. And at this point, I want to also thanks to everybody who got me to this point in the development of this cure, including feedback from Justin St. Juliana, George Middendorf, Joya Mukherjee, who is the cure queen in our department, and she helped me a great deal thinking about the cure, and the whole FMN cohort that semester. And also thanks to Teresa for inviting me to be here today as well. Okay, so more background. I'm at California State University, Sacramento. We are situated it's there circled in red, and we are right on the American River, one of the largest rivers in California. And it's literally a stone's throw from our building. I look at the river from my office, and the American River is part of the whole American River watershed, which is on the kind of the west side of the Sierra Nevada, west of Lake Tahoe, as you can see here. And so that includes a whole watershed, and we're just downriver from Folsom Lake. And all along the river is the American River Parkway. And this is a 32-mile greenbelt along the American River. And there are 82 miles of maintained trails. There's 8 million visitors annually to the parkway. So it's well used and well visited. And it includes many different kinds of habitats, of course, riparian, but also oak woodland, grassland, and so on. And it's quite diverse, the parkway. There are 40 species of fish, over 30 mammals, of course, hundreds of plants, over 200 species of birds are also found along the river. And I wouldn't be a good ESA member if I don't show you some of the wildlife along the American River. And it spans bald eagles and turkey, of course, and deer and beavers and a number of species of snakes and so on. A little bit about our student population. I am at a primarily undergraduate institution. We have over 31,000 students, and we are one of the most diverse campuses in the the whole nation, but also the fourth most diverse in the Western US. We've been a minority serving institution for a number of groups, as you can see, for many years now. And a third of our students are the first in the family to attend college. And in general, our students tend to be a little bit older. A lot of them work full time, a lot of them have families and so forth. So that's kind of the makeup of our population. And when I started thinking about where to take my project for the FMN, I, of course, had in mind this general ecology class that I teach. It's upper division, and it includes a lab. And I thought that would be the place to really incorporate some kind of project, including the human-environment interactions. Now, I gravitated towards Cure for a number of reasons. And Cure, first, essentially, like what I was already doing in the class, even going back to when I started as a faculty member, was pretty much a cure. And then for over a decade, our department has developed many cures that are embedded throughout the curriculum. And so it seemed like a easy jump to develop one and develop it further for this ecology class. So cures, if you don't know what they are course-based undergraduate research experiences. It's a equitable way to provide research experiences to undergraduates. A lot of of course, as I mentioned, a lot of our students are, have busy lives and don't have time to participate in research in a faculty's lab. And furthermore, as being an primarily undergraduate institution, we have heavy teaching loads. And so we don't have all the time in the world to mentor and have students included in our research. So this is a great way for everybody to meet halfway to provide the research experience, this genuine research experience for students in the classroom. And so they can engage in the scientific process from really conception to communication, and it's incorporated in the classroom. So, and I already talked about this being a general ecology class, upper division. Oh, I should point out that the students are quite a mix of what major they're in. We have probably almost half are environmental studies students, half are biology, and there's a segment of the biology students who are kind of on the pre-med track, and so they're not all ecology minded, and everybody is coming in with different experiences in the classroom, especially since we have these two different majors that are taking the course. So Amanda did an excellent job going through the 4DE net framework. And so I'm not going to go into detail, but just what I have listed here are the four concepts, and I've indicated kind of what parts of those concepts do I emphasize in this cure. And so, of course, with the ecology concepts, it's kind of all levels of ecology. The cross-cutting themes are temporal, spatial scale, and of course, with the practices, it spans natural history to fieldwork to the quantitative end of things, the critiquing and the collaborations, and then finally, with the emphasis of the FMN and this cure is the human-environment interactions. And the key point here is that humans shape and manage ecosystems and environments. And I have pictures of just different aspects of the environment along the American River as it pertains to this HEI. So this includes invasive plants. Of course, we experience droughts periodically, habitat modification, which I'll talk about in a little bit more. And then, of course, there's habitat restoration, which I'll also talk about a little bit more. Oops. Okay, so here are the student learning outcomes and the assignments associated with those learning outcomes in the class. Initially, in the beginning of the semester, I'd break up students in the lab into groups of 3 that they will be working with for the whole semester. And I usually let the self-assembly of those groups of 3 happen. And from there on, we are touching on this cure for the rest of the semester. So really at day 1, they are experiencing the different learning outcomes and assignments associated with the cure. Although I should point out that I do have other exercises throughout the semester. So this isn't like this doesn't incorporate the whole lab. But they include things like critically assessing scientific literature, and the assignment is article anatomy, which I cover over a couple weeks. The applying ecological concepts, and there's like an HEI walk along the river, a cure proposal, HEI reflection at the end of the semester. They also design and conduct a study. Of course, that's the whole point of this cure. They manage and analyze the data. A big part of this is collaborating with others and on the research project and dissemination. So, and then of course the communication of, the communication of their research. So in the beginning, we're starting to build up their skills and in the development of this cure. We do a class project together where we collect data and analyze it, and it turns into a paper. But for the most part, they then are starting to work on what their original research is going to be. And all this is guided by me. I give them the flexibility to kind of go where their interests lie, but I make sure that it's something that's doable within the class and the class period. And also it's something interesting and relevant to the course. Whoops. Okay, this is just pointing out that that we cover this throughout the semester. Okay, so what are the products? And these are the products that are found in cubes. So I won't belabor it, and I've kind of covered all this. I just want to highlight the proposal worksheet. I've broken up the proposal for their project into 3 units where they're kind of taking it piecemeal and building on the assignment and the development of their project. And I'm really proud of that aspect. And Joya was really a great help in thinking about it in that way and preparing it, instead of like just this one proposal that they turn in at some point in the semester. There are landmarks that kind of help guide them to that finishing point. All right, here are just some examples of, so the FMN was in the fall and I implemented this in the spring. And here just an example of some projects. One was, and you can see in the upper right-hand picture, it just so happens that they were doing work on the levee right across the river from our campus. Building it up. And so there was levee construction that had just finished. And so one group did a project looking at levee construction effects on soil and vertebrate communities, plant community structure along an environmental and human disturbance gradient was another project. Distribution, abundance, and life history of an invasive species along a moisture gradient. And American River water quality and invertebrate abundance associated with salmon and levee restoration. So there not only was there work being done on this levee, but there were several locations along the river not too far from us where they were doing salmon restoration, well, building up the habitat for to encourage salmon spawning in certain areas of the river. So we're just reflecting on all that semester of implementing this, and just thinking about this widely, because I'm sure not everybody is situated right on a river or and so forth, and may not have access to this. But I really think that they're just the structure of this cure and the approach of this cure in the context of this human environment interactions, I think could be adapted to many other ecosystems. And if anybody's interested in discussing that, I'd be happy to discuss that further. But I think it goes beyond just aquatic environments, riparian environments, and can include many different kinds of systems. Possible add-ons to the cure are like a electronic lab notebook. I may try that in the future. One group wanted to do a vlog and did, and so that might be something that you may want to incorporate into the cure. I'm not sure whether I will, but we'll see. And then, of course, long-term data. So that might be something that could be built into this, where we are collecting data that may be of use to the various agencies in California. And we could build up a data set that I think could be, really useful and relevant to both students and the community. Also, another reflection I had on is that students, as I mentioned, they come from different departments. Some are even transferring community college. So people are coming in with really a wide range of quantitative and communication skills into the class. And I found that to be quite challenging to deal with. And so I'm still toying with how to approach that aspect to make sure that everybody kind of is at a similar place and can contribute equally. And speaking of equity within groups, the age-old problem of everybody contributing to the project and all aspects of the project are always a challenge. They always have been, and so I include many different approaches to that, but that's still an ongoing issue. And I just want to point out that what I published, this Heal Cure, it's a living document, and so any feedback on it would be greatly appreciated. And I will certainly be updating it as I teach this class more and more. Here's my information, my website, and I believe in the email that they sent out, there was the link to the CUBES publication. But thanks for listening.
Thank you so much, Jamie. Thank you. And I think we have about 5 minutes left for any questions that anybody may have for Jamie.
I have a quick question. So those research projects are very interesting. Do you anticipate any publications or micro publications arising from those?
That would be really fun. I do. I'm not sure whether I don't know. I'm not anticipating. I don't hold my breath. But what I do envision, there's a lot of opportunities for students to present their research on campus with different like research symposia. In fact, we just had one this week where students can present posters on their research. So I do envision hopefully that students would be, open and willing to present in those. The problem is, the timing. And so, for example, like those projects were last spring. A lot of people graduate, and then, or the next semester, it's just kind of like they've moved on with their lives and their programs. So yeah, I mean, I hope to somehow have that included. I'm not sure whether like an actual paper, but who knows? There in the past, there has been some projects where I feel like we really could have turned them into project, but that's a paper, but that has its own challenges of having everybody on board and then time and effort. So yeah, we'll see. It's a great idea.
Thank you, Amanda. And I think James has a question.
Hey, Jamie. Good
Hey,
to see
Jay.
you.
Good to see you.
I was excited to hear your lesson, and I'm excited to share it with the River Field Studies folks because I saw the title and thought probably relevant for lots of that network as well. This isn't a question so much as an idea. I was really excited listening to your talk and being in Richmond, which is on the James River, very close. We're kind of like an East Coast parallel, state capital, diverse school. We're kind of like the eastern version almost of Sac State. It'd be fun to think about doing a distributed, like doing a coordinated class where we run the Cure with a group here on the James and a group with your students on the American, and then do some sort of a virtual symposium. Getting back to that idea about sharing out the results, we could have maybe students presenting across the country with some shared ideas. That could be, I mean, it's a little bit pie in the sky, but if it lines up that I'm teaching at the same time you are, we should spitball around that.
Absolutely. I would totally be up for that. That would be a lot of fun. I think that's a great idea. And yeah, we should definitely talk about it. It would be challenging, but I think we could do something, and it would be great I think it'd be great for the students too to see these different perspectives and other projects from groups across the country. Yeah.
Also makes me wonder, I wonder how many, probably a bunch, river capital cities with universities there are across the country.
Yeah.
And doing some sort of a undergrad network where, we all do our river focused cures across, because you guys have the Sirius program, right? Where you've embedded the American River really integrally into your whole undergrad STEM program.
Yeah.
Maybe not quite as big as that, but it'd be cool to have students from across the country, but maybe you and I should try it first, see if it works.
Yeah, no, that'd be great. And Sirius is a very different, and again, that's, it's a wonderful program. For those of you who don't know, I totally blank on the acronym, but it's built cures, and I mentioned this early in my talk, that it's kind of a bunch of built cures and scaffolded skills that are embedded in our whole curriculum, and it's all surrounding the river. And so we have micro classes that do microbial diversity of the soils along the river, and and so on. But yeah, this is a little bit different because it's across. It's here. It's the spatial variation, and with everybody being ecologists and focused on ecology. I think that would be, yeah, really that's a great idea.
Teresa's got a network for us to look at in the
Oh,
chat
does she?
area.
Yeah,
Thank you.
just a plug for the EREN, Ecological Researchers Education Network. They actually have methods to help people who want to do that kind of cross-country type of collaboration, for student research. So you might want to check that out.
Yeah, thank you. Dan, thanks, James, for bringing that up. It's a great idea.
Thank you so much, Jamie, and thank you, James, and thank you, Amanda. I think we have about 5 minutes left. If anybody has any last-minute questions for either Amanda or Jamie, we can open the floor for maybe 1 or 2 more. No, okay, looks like okay.
That's one.
I think the Christoph.
Hi, everybody, and thanks to both of you for the talks. They're really good. I just wondered, I assume that there's a lot additional material that students need to read through and know when you do the field work. How do you manage to get that in addition to the, I want to call it regular readings? I noticed that I'm in Canada, so we have only 12 weeks. I saw that you have like a little bit longer. Do you have any hints or tips as to what might be most important and useful?
Yeah. So I get them started early. And so, in fact, that article anatomy is, so I like killing many birds with one stone. And so like that is focused on some kind of riparian habitat, human environment interaction. So they're already getting started then. And the other projects are also, I'm current and I have them read papers, and then as part of the proposal, they have to also read some papers. And so it's not overbearing, I guess. Prior to them coming up with the hypotheses, they're reading, 3 or 4 or 5 papers before that, and then of course it's all guided by me as well. So whatever direction they start going in, I suggest papers that they can read and look at as well. So you're absolutely right that them having the background is a challenge, but it's already embedded in the class, and our whole curriculum, they're starting to read papers in their first biology class. So they're not brand new to reading papers to begin with. And this is an upper division ecology class. So they've gone through many classes where they've been reading papers and critically assessing them. I see.
And that was going to
happy to talk further, but I see there's Porsche.
Yeah, one more.
Porsche. I have a question. So it kind of is similar to Christoph's question about, the time, because I know like when Cure courses, when you like with microbiology, for example, it doesn't take long to set up those experiments and generate an amount of data where you can actually have a little bit more analysis. I mean, to conduct statistical analysis. But with field work, you have to, have multiple field days, and the weeks being down to 15, maybe 16 weeks in the spring semester, getting them started, which can take about 2 or 3 weeks. They only leaves you like maybe 3 weeks of field work for them to actually put together a product. So what would be your recommendation on how to, because I'm interested in setting up Q courses myself with integrating field work into it, but my concern is the data that can be generated, the little bit of data that can be generated for them to actually put together a product.
Yeah, that's a great point. And again, part of it is just me providing feedback for them to develop a project that will only require a few weeks of data collection. But it does get really challenging, and especially for the students who want to do something with invertebrates, and because there's not only sometimes extensive field work, but bringing the specimens back to the lab and so forth. And so, and then there's times of the year, right? Like the fall is really, really challenging to do these field projects compared to the spring. So there's also that seasonal component. And it's just a matter of like really providing that feedback and oversight with the student projects to make sure that they are doable within kind of that time frame. And I do have, there's some buffer, right? So I have like 3 or 4 weeks where they can be collecting data, but there is still potential. Sometimes I've suggested students start even earlier on their project, and then sometimes there's a little bit of buffer on the tail end to collect more data. But it's just one of those challenges in the constraints of the course and time, to provide the feedback for a doable project within that time period.